finish up today by looking at the basal nuclei and limbic systems. And then we'll look at the three major components of the diencephalon. And then we'll be ready for the midbrain brainstem structures on Tuesday. Don't want to tell anybody you're here. All right, so this view, and let me just kind of make the contrast here a little bit better. In this view, we can see a continuation of that pattern of the C-shaped form, and we're talking about the caudate nucleus. So we had these structures that start with the letter C, cingulate gyrus, caudate nucleus, corpus callosum, that all seem to contribute to the same C shape. This medial structure right here in the most midline, you can't quite see it, is the thalamus. So some of you are thinking, we had to read clay today because you see some of the clay structures. I was going over, here's my clay visual here. I was showing some students during the lab how the structures we're talking about today are associated with the lateral ventricles. I wish we had enough of these made out of metal for everyone to use, but we will use the paper covered countertop because these are too fragile and we don't have enough of them anyway, but I'll kind of show you on this today and on Tuesday. So notice that these structures are gray matter inside the brain. All right? That's what a nucleus is. So a gray matter cell body's outside the central nervous system is a ganglia. Older terms for this, we'll call them basal ganglia. You may read various texts, the basal ganglia, but we're going to describe them as basal nuclei because it's collections of cell bodies or gray matter inside the central nervous system. So as we look at this structure, and this I'll show you over here with it labeled. Okay, I just wanted one a bit close up. So if we look at the structures that form the basal nuclei, we're on page 72. As we move from center out, we have the thalamus here, all right? And then lateral to the thalamus is going to be the globus pallidus, the putamen, and then finally the caudate nucleus. That, along with the part of the midbrain, substantia nigra, and the subthalamic nucleus, for most people are the structures that are listed that form the basal nuclei. All right, so before we go any farther, why are we talking about the basal nuclei? Why are they important? And anybody in here doing Parkinson's? Okay, this is your day to listen carefully. So I was trying to come up with an example, an illustration of how the basal nuclei work, because we know from what I talked about on Thursday and on Tuesday, that when we decide to do something, frontal lobe, we have the precentral gyrus with our upper motor neuron, which is going to send its neuron. It will cross over in the brainstem. It will go down to the spinal cord, and then the lower motor neuron will go to the skeletal muscles. And that's what we need. We might have our premotor cortex that plans the sequence of what we're going to do. But I left you with the impression yesterday that if I decide to extend my arm, this is what gets activated. The upper motor neuron, which activates the lower motor neuron, which activates the muscle, and I extend, or muscle cells, and I extend my arm. All right? So why have a basal nuclei? Why don't we just control it? Well, these upper motor neurons can be activated by a whole series of varieties of other stimuli and other electrical currents in the brain. So we need some kind of a funnel to control what stimuli actually get to those upper motor neurons and have some other level of control. So the closest analogy I could come to is that the basal nuclei function rather like the starter on a car. Your muscles would be the engine, all right? And they would work fine. The starter starts the I don't know cars, so I imagine there's an electrical current that goes from the starter to the engine to get the alternator, okay, thank you, to get that going. But if I want that engine to work, I have to turn the starter, okay? And so the basal nuclei is your arm, is that moving forward and actually turning the ignition. That's what the basal nuclei do. Because when we have disorders with the basal nuclei, the ignition is actually never turned off, or you sit there in the car, so like Parkinson's disease is an example, where the driver's sitting in the car, the engine works perfectly fine, there's gas, but the driver's just sitting there, his hands on the key, or in my case on the Prius you have to push the power button, and it just, no matter how much that person wants to turn the key, they can't. They want to move, but they just can't get that starter going so that the muscles will move. And that's what the basal nuclei is doing. It's turning the key. It's getting the starter going, all right? And so it's a complex series of electrical currents, neural circuits between these structures. I'm going to go over, that's the slide that scared all of you when you were coming in. And you didn't see it before because I made it up last night, but I just put it up on D2L as you were coming in. And so I'm going to go over that with you, but we're going to spend most of our time talking about naming these structures and identifying them in the brain. But that's what I want you to come away with, is that this is our kind of subconscious control of whether the muscles are going to work or not work. When the basal nuclei are not healthy, we can have either extreme of the problem. We can have Parkinson's disease where we don't have enough movement, or we can have something like hemiballismus or Huntington's chorea or hand tremors where there's too much movement. Okay, both of those can be disorders of basal nuclei. So these are the components that we find within the cerebral hemispheres that would be part of the telencephalon. The caudate nucleus, the putamen, and the globus pallidus. Whereas the substantia nigra is going to be part of our midbrain, what we talk about on Tuesday, and the subthalamic nucleus is part of the diencephalon. So let's take a look at how this fits in with what we are, I was going to say no, but you may not know it until next week. So what you're becoming familiar with. And keeping our colors correctly here. So the most medial structure is going to be our thalamus. Okay? And the hypothalamus will be coming in like this. So that's the thalamus, and I'm going to draw it kind of as a heads-on view here, what we see. So that would be thalamus. All right, so this is one side. This would be looking at somebody and seeing the left side of their brain. Okay. To keep that in better perspective, I will draw in the third ventricle. Okay. So that's kind of cutting right down through here. Kind of with me on what we're looking at. This would be like a coronal section of the brain. Okay. So our longitudinal fissure coming around like this. Our lateral sulcus. I don't want to bring it quite that far. All right. So now between the thalamus and our caudate nucleus, we have the globus pallidus. So let me refer to this model that I was working on earlier today. So on our ventricle model, this middle piece right here was our third ventricle, right? Okay. So this is going to represent the thalamus. And the thalamus forms the lateral wall of the third ventricle. So I put that in there, forming the lateral wall of the third ventricle. Now forming a rather pyramidal structure. I have with its point facing towards the thalamus. I have the globus pallidus. We're going to put that here. And that we're seeing the wide part with the point kind of pointing in towards the thalamus. That would be this structure. Okay. And then over that, we have the putamen. This is a front-on view. This is a lateral view. You guys with me? We're building this up. All right. So this orange structure right here is the putamen. So it's kind of a flat pyramid. So no matter which way I cut it from the top, from the side, it looks like this triangular shaped pyramid. So I'm going to place it on my thalamus like this. Again, we're going to be doing this about the model, the ventricle structure, on Tuesday. Okay. Now there is actually a gap between the thalamus and the globus pallidus, but I have to stick it on there because I can't suspend it in air. So the red is the thalamus, and that orange structure is the globus pallidus. Okay. Should you guys And now I put the putamen over that. And the putamen is this second structure here, and it's going to be this kind of teal structure here like this. All right, so I'm not going to walk around again, but that's the teal structure, so it's more flat. Yes?
You have it listed the globus pallidus. Is that just two parts?
There's an internal and an external. So the globus pallidus has actually
Oh, okay. That's why I'm getting confused. Seeing three structures.
like that.
Okay.
Okay. And then you have this large, the putamen kind of covers over that, and it's more flat. It's also called the lentiform component because it looks like a lens with those two structures together. Create a lens-looking structure. Okay. And then the next structure is actually now this C-shaped structure. So it has the head. So the lentiform, the putamen is now considered to be part of the globus pallidus. In the past, it's been considered separate, but now it's considered to be part, so they have it the same color there. And notice what I do with this caudate nucleus. It again has a C shape. The large head is anterior, and it actually bulges into the lateral ventricle. So on our brain models, when you take the top off of the half that comes apart, on our brain models, when you look at that, you can actually see the head of the caudate nucleus bulging into the lateral ventricle. It then curls around into the temporal lobe, and the inferior horn has its roof formed by the tail of the caudate nucleus. All right? So head of the caudate, body of the caudate, and then as we move into the temporal lobe, we get inferior horn. And the tail of the caudate forms the roof of the inferior horn. So body is the lateral wall of the anterior horn. Head is the lateral wall of the anterior horn, and tail is the roof of the inferior horn. So we'll see this when we do cross section. And you'll be modeling this again on 2D.
Tail is the roof of
So head, body, tail of the caudate nucleus.
чего? Horn.
Head,
Inferior horn.
body, and tail. And there's the thalamus, medial, globus pallidus, and putamen lateral. And then the C-shaped
Putamen lateral.
caudate nucleus going around it. So if I draw that now on the board, so notice how that at times when I make a section, a coronal section through the brain, I'm going to be cutting the body and the tail at the same time. When I'm doing that, I'm in the third ventricle. All right, so that will help me see as I'm going through cross sections. I'm going to show you some slides of that in just a moment. But you can see this relationship. So when I see the thalamus, all right, there's the inferior horn. The lateral horn. Here's the inferior horn. So when I see the thalamus, and unfortunately my colors here are not the same, but I would be at this point here, and so I'm going to see the body of the caudate there, and the tail of the caudate here. And the reason is because of that C-shaped pattern that's occurring. So here's another view. I think this is from Netter's. He has one similar to that. We'll talk about the amygdala, that little circle in the front, when we get to the limbic system later today. So if we look at this is similar to what I drew over here on the side. So this is the same picture from Netter's that I showed you for the hippocampus yesterday when we were talking about the temporal lobe. And the hippocampus forms the floor of the inferior horn. So here would be the thalamus. And again, I know this is thalamus because I'm in the third ventricle. Okay, and the thalamus forms the wall of the third ventricle. So this is thalamus. This is the lateral ventricle. Here's the body. Here's the tail. Excuse me. Here's the tail. Here's putamen. And here's globus pallidus. Okay. So that's a very common view of a coronal section of the brain with the third ventricle in place. I'm going to show you a series, though, where we start in front and we go posterior. We're going to start in front of the third ventricle and end up behind the third ventricle. And if we start in front, we're going to move into the head, and it's going to be huge. And then we're going to see the thalamus, and the head's going to get smaller as we get to the body. And then we're going to get back and get body and tail and third ventricle and keep moving back posteriorly. So it helps to keep this type of a pattern in mind as we look at these views here. So this is an MRI. Very similar pattern. Lateral ventricle, third ventricle, thalamus and hypothalamus, putamen and globus pallidus. It's hard to see the body and the tail in this view. Now we have plastic sections of these coronal sections of the brain, plasticized sections in the back of the room. In your lab manual, there are black and white printed pictures with labels. They are in color on D2L. Okay, they are not on the black and white. This becomes sometimes hard to see, but I have labels, and the same pictures are in color on D2L. The pictures that I'm showing now are ones that I found last night. Some of them are, the ones I showed you aren't labeled, but some of them are labeled on that site, and I can put that up. I want to go through this with you. We're going to do this multiple times over the next few days. Let me just turn the... Okay. Now, I'm going to go slow, because we're going to see this over and over and over, but I just want to remind you what our view is that we're looking from the front. All right? So let me get a whole ray, just to kind of half of a whole ray. So when I section right here, I am cutting all the way through the longitudinal fissure because I haven't reached the corpus callosum yet. So on this view, A means it's the front side of the slice of bread as I move from front to back, and B is the back side of that same slice. Okay? So you can see the longitudinal fissure goes all the way through. As we look through it, I can see some structure here. And when we look at the section, the bottom of the second slice, that's my corpus callosum. And if I look on the sagittal view, it's quite fat there because it bends under itself. Remember when I took that yellow paper yesterday and I bent it under? So that is a quite thick part of the corpus callosum. And so the front of the brain there is going to be thick. You can see a gyri and sulci, and short association fibers would be doing this, right, within the same frontal lobe. I couldn't get the 1B to show up, or the 2A, but still we can see we're still in the frontal lobe. Corpus callosum, taking frontal lobe fibers to frontal lobe fibers on the opposite side. All right, and then in the next slide you're going to see just the beginning of the lateral ventricle. So we're seeing just the very, very tips of the anterior horn of the lateral ventricle on each side. We don't have enough of the lateral ventricle to really see much of the head of the caudate. We'll pick that up as we move more posterior. And here we are. So this might look like thalamus, but we're not anywhere close to the third ventricle yet. This is still the lateral ventricle. Notice how it's bulging into the lateral ventricle. So it's that head of the caudate nucleus bulging into the lateral ventricle. And this surface right here is actually the caudate nucleus, that gray matter. Now we're into the temporal lobe. You just picked up part of the temporal lobe. And here's the lateral sulcus. We're not far enough yet back to see much of the insula, none on this side. But here, the head of the caudate nucleus is so large it's almost collapsing the lateral ventricle as it bulges into that. This is still corpus callosum and longitudinal fissure. Look how huge that is. It looks like it's the head, but if you look closely, this is the head of the caudate right here, and we're starting to pick up the putamen right here. Kind of see these little striations, just barely. That's why the caudate is called the striatum. The putamen and caudate together are called the striatum because they look striated. There are myelinated fibers that go back and forth between the two and give it this striated look. Now you can see that a little bit better. Okay, right there. We're still not in the third ventricle, picking up the optic nerve here. And you can see the insula here with the lateral sulcus, insula, and the lateral sulcus, corpus callosum. Just barely starting the third ventricle here. Okay. So here is caudate, putamen, and there's a little bit of a light gap there. This is globus pallidus. More of that. Caudate, notice how this is getting smaller. So we're in the body. Putamen, globus pallidus, And in our next slide, we should see the thalamus. There it is. Here's our third ventricle just starting, and here's thalamus. Right here, and right here, because caudate's up here. Okay, we're also starting to get just a little bit of our inferior horn. And caudate tail, the caudate's up here. A good putamen and the two parts of the globus pallidus. This is septum pellucidum, which is the membrane that hides the lateral ventricle. So if you look on the half of the brain, there's this thin membrane. Well, it's not a membrane, it's plastic. But on the sheep brain, we're going to have to cut that, and when we do, we'll be inside the lateral ventricle. So it's just barely that thick. All right? So body of the caudate, tail of the caudate, all of this is thalamus because we have the third ventricle. Inferior horn, and you can see how the temporal lobe folds up and pulls choric plexus with it into the inferior horn. And this is part of the pons, and this is the angle. We're getting part of the pons in the midbrain. So see this dark layer right here? Substantia nigra, which is part of our basal nuclei set. You can see it a little bit better here. Here's hippocampus. Here's body, tail. We're starting to, we've lost our globus pallidus, but we still have some of the putamen. Third ventricle. Now next time we look at this, I'll talk about fornix, which is part of the limbic system we're going to end with today. Still see the inferior horn, still see part of the body, still see thalamus. We're moving down into the brain stem. Part of the cerebellum. And we're going to get back in here. We're starting to move into the inferior horn. Here's fourth ventricle, with the medulla and the pons. And finally, we're almost out the back end. All right, so we'll look at this again. I'll show you, use the pictures as well that are on D2L. But I kind of wanted to focus on caudate nucleus and putamen and globus pallidus as we look through these structures and start to get used to what they appear like in cross-section. So, the components of the basal nuclei, this gray matter that I have on the board, receive a lot of inputs from all over the brain. They receive inputs from the cerebral cortex, both motor and sensory. Okay? I'm going to give you more detail than I'm going to ask you to know, just to kind of give you that information and back up from it. So it's like we're walking through the forest and then we're going to back up and you just need to recognize the forest as far as the total information is concerned. It's going to get information from the thalamus, from the subthalamic nuclei, that just means some cells, group of cells below the thalamus, and the substantia nigra. Now these two structures, three structures here, we haven't even talked about yet. All right, we referred to them, but we haven't gotten to them yet. We'll do most of them today. So, I'm going to put this pattern up on the board. I just made this last night. I've struggled over the years to find information that clarified what the basal nuclei do, but unfortunately people don't know exactly how it works. And so the information changes, and they change their minds and so on, and it can get really complicated. But this article I found last night was probably the best one I've found in years. And I used its illustration to draw this on PowerPoint last night. So, may I erase the board? Are you guys all right with that? So first of all on the board, I've drawn the cortex, okay? It's going to be the motor cortex over on this side, but just general cortex. And then, get myself room here. This is the caudate nucleus. And the putamen, which are now considered to be one structure. And the globus pallidus. The medial and lateral, or internal and external, as it's called. and then part of the thalamus. Most of the thalamus is sensory in function, but a couple of the ventral nuclei, ventral motor, so a couple of the ventral nuclei are going to be motor in function. I'm going to add in the subthalamic nucleus in just a moment. So over here, I want to use a different color. Over here is represented the precentral gyrus. All right. So this is going to be the precentral gyrus, which remember is the upper motor neuron that we talked about yesterday. That's the descending motor pathway. So this is going to be upper motor neuron. And this is going to go down through the brain stem and cross over to the other side of the spinal cord. So from some area of the cortex, all right, premotor area, frontal, sensory, a variety of locations, we get an excitatory neuron. And I'll explain what that means in just a moment, because that concept itself is in our next unit. But from the gray matter of the cortex, a neuron descends to the caudate nucleus. It releases a neurotransmitter, which is excitatory. And what we mean is it is more likely to cause the neuron it synapses with to form an action potential. So remember, the action potential is that electrical current that is traveling along the axon. When it gets to this neuron, it causes it to release neurotransmitters. Those neurotransmitters can bind with another neuron and can cause them to do the same thing. So when this neurotransmitter binds to this neuron, it makes it more likely to respond by forming an action potential. So this is considered to be excitatory. It's more likely to cause this neuron to fire than to inhibit it from firing. So it's a stimulatory effect, all right? So that's why I have the plus there. Because the result of the glutamate, which is an excitatory neurotransmitter, the result of glutamate binding to this is going to make it reach an electrical current. It's going to respond by creating an action potential. So that's why I have a plus. This cell is more likely to fire. When it fires, it sends a signal to the globus pallidus. Now you'll notice that I have a negative by this one. Try not to get too much into the neurophysiology yet. This cell will release neurotransmitters, but the effect is to make the inside of the cell more negative. All right, so how do we do that? There's two ways we can make the inside of a cell more negative. We can make the inside of the cell more positive. Typically, and almost always, we do it by adding positive ions. So sodium ions go in through, remember the ligand-gated channels? Sodium ions go in through those channels, and the inside of the cell becomes more positive and a current forms. If the current's strong enough, it travels down the axon. All right? How do we make the inside of a cell more negative? Exactly. We can lose potassium. The channel that opens, instead of being for sodium, could be a potassium channel. And if we're leaking out potassium, then we're not going to make a positive current. Okay, the inside of the cell is going to become more negative. It's going to be inhibited. Another way of doing that is to add chloride ions, which have a negative charge. You can do either way. That's physiology. I'm not going to test you on that on this segment. I'm just kind of illustrating how this works. We'll learn that in our next segment. So when this neuron fires, it binds to a neuron, and it makes this neuron less likely to fire. Does that make sense? It's going to make the inside of this neuron more negative, and therefore this neuron we'll just make a statement, it won't fire. It won't send an action potential when it becomes more negative. Its neuron goes to the motor part of the thalamus. And its effect is also to create a negative potential, to make the next neuron not likely to fire. So let's stop here before we go on. Two negatives make a positive. So if this neuron makes this neuron not fire, and if this neuron is a negative and it doesn't fire, it's going to make the next neuron more likely to fire, right? So if I have this cell body, and if I add a lot of positive ions to it, it's going to fire. Okay? If I add a lot of negative ions to it, it won't fire. So let's say I don't add those negative ions and other neurons come along and do add positive, then it will fire. So what I mean by a double negative is if this one is negative and it keeps this one from firing, if the normal role of this one is to keep the next one from firing, then if this one is active, it's going to stop that next neuron from firing. But because I inactivated it with this one, the effect of that double negative is this neuron that's going to activate my upper motor neuron will be active. So like just a whole bunch of ways to control this. So the net effect of firing this neuron is to cause my upper motor neuron to fire. We went through all of that just to turn on the ignition switch for my upper motor neuron to fire. Okay? If this system doesn't work, will my upper motor neuron be more likely to fire or less likely to fire? Less likely. That's the essence of Parkinson's disease. Okay? So this is called the direct pathway. It activates. It is going to be, so when this is going to send the neuron activation down my my upper motor neuron. It's going to get to the spinal cord and it's going to go to my muscle. And so it's going to increase motor activity. Now, it wasn't quite as complicated as it looked. Just remember, it's just a whole bunch of extra steps in there that causes your muscles to be able to work. Okay? All right, now we have the opposite effect. We have to be able to make our muscles less likely to work. And so we add the subthalamic nucleus. Now before I go further, let me just show you where the, this is going to take a while. Let me just show you where the subthalamic nucleus was. Here's the thalamus. Subthalamic nucleus is just the little one right underneath it. Okay, it's not something you have to identify in lab, but I found it helps when you're learning something to be able to picture it. And that's where it is, with the substantia nigra being this structure right here. Okay? So for the subthalamic nucleus, we use a different part. So this neuron is going to go to the more lateral or external part of the globus pallidus. It also has a negative effect, just like the first neuron did. And it synapses with a neuron that also has a negative effect, but it goes to the subthalamic nucleus. The subthalamic nucleus has an excitatory responds. So we just put in an extra step here. The net result is a double negative is a positive, right? So here's our double negative. Our positive is to excite this one, which causes this one to inhibit this one. So our net result is we block activating the upper motor neuron. I will be happy to repeat it 3 times if necessary. On our first pathway, our double negative was a positive. This green neuron was allowed to be excited, okay? Because we inhibited the inhibitor, essentially. We inhibited the inhibitor, which meant we excited. This pathway activates the inhibitor. So if you think of this neuron right here as the inhibitor, here we're inhibiting the inhibitor, and our muscle works. Here we're exciting the inhibitor, and our muscle doesn't work.
Then the neuron will be red.
So this one is, well, this is the inhibitor.
Right.
So this one excites it. So when this releases it, negative ions go into this cell. It doesn't work, and so this cell is less likely to fire. Okay? So this pathway increases motor activity. This pathway decreases motor activity. This pathway inhibits the inhibitor. This pathway activates the inhibitor. Yeah.
Sorry. I lost what you said about the first green neuron. Why does it turn red?
So it doesn't turn red. The color red means that this releases a neurotransmitter
That will.
that causes this neuron to become more negative and not fire. Okay? So this one is green because its neurotransmitter makes this neuron active.
So the second neuron is active.
So when this one fires, this one will fire. It will send an electrical current along its axon, and it will release neurotransmitters
Specific neurotransmitters
that will bind to this one, and it will cause this cell to not fire. Okay? This cell also releases neurotransmitters that keeps this cell from firing. So when this one inhibits this cell, and this cell doesn't fire, because it can't. Remember, if I let negative ions in here, it won't fire?
Right.
That's what these neurotransmitters are doing.
Okay.
So let's start back again. This cell fires. It opens sodium channels in this cell. Okay? And this cell, it does the same thing for this one too. So both of these are going to be more likely because they're going to become more positive. Therefore, sodium channels are going to be open. As sodium ions flood into that, that creates an electrical current that is propagated down this neuron. And at its axon terminal, it releases chemicals too. These chemicals, however, cause this cell to become more negative.
Right.
So it doesn't make a current. And it doesn't send a signal down its axon. If it doesn't send a signal down its axon, it doesn't release any neurotransmitters. If it did, its effect would be to make this cell more negative, and this cell wouldn't work. That's the double negative I'm talking about. So the most, so let's say I have this cell fires, it makes this cell fire, it releases when this cell opens, positive ions come into the cell. Those positive ions travel through the cell body and open voltage gated channels, and our current travels along. Okay, that's a positive effect. All right, now let's say that when this cell fires and it releases positive ions, and when those ions, let's say, but when it releases ions, those ions are negative.
It's a different neurotransmitter.
So if those ions are negative, I'm not going to make a current.
Right.
And so this neuron will not release any neurotransmitters,
So the next neuron won't fire.
and the next neuron won't fire.
With the next neuron not firing, it's going to cause the following one to fire.
It's going to allow it to fire from other sources.
Okay.
So this one, because this is a negative, it stops another negative from working, therefore this one will work. This is a negative, all right? This one's a negative, it stops this negative from working, so this is just like this one here. This one now will fire, and when it fires, it turns this one on, which makes it release,
Which makes that not.
which makes this one not fire. So we have two situations. This one either fires